The only person who is not here, I guess, is Maylin. I have sent her an invitation. Give it a couple of seconds before, couple of minutes before she joins. On the other hand, I can't remember whether she asked to be excused because she had to go somewhere. Okay, all right. Well, we'll see. Let me just check. Okay, she was going to come in today. She's not. I'm not quite sure about why she's not here, but I've asked her to request to join. I'll start at about 5 minutes past. Hey Tim,
How you doing this afternoon, sir?
doing all right?
Okay, I'm gonna ask a favor, and I hate that I have to ask it. Uh, I don't know if I'll go back to work, but for Money's account, will you make a copy of the identification card through Passport for me on this paperwork, please?
Okay. Passport is required. Is that correct?
It is not. This is strictly for mine because we're traveling to Colorado and very active ICE traffic in Colorado.
Okay.
So that's how come I hate asking for it, and I'm here to protect Travis because okay.
Okay. So this is not a problem. Uh, do we have an issue? His health thing is it all in place? Because he kept saying that it needs to be done online, and then I said, okay, come sit with me, and we never got around to doing it. So
Yes, you should have gotten an email from Kim Shaw
okay,
on Monday.
okay.
Did you get that email?
No, I never did.
All right, let me go back and get that sent to you,
Okay.
and on the 6 month
Mm-hmm.
on the
My detail baby knowing the
okay.
girl you can also kill it.
On uh, the Part C of that form that we had done.
Yes.
You'll have to scan in, get it in there, copy your insurance card,
Okay. Okay. All right. Not a problem.
Okay,
We will.
sir.
Okay. All right. Take care. I'll do that.
Bye.
Thank you. Bye. Sorry about that. Sorry about that. Things never never seem to end. Okay. Think we have a quorum. Welcome, Maylene. Everybody's here. Let us begin. Okay. So first starters, can everybody hear me? At least one person raise your hand. Okay. Thank you, Michaela. All right, let us then begin. So today, this series of lectures, which might go into next week, is about about sequence analysis. Okay, so this is about the kind of the classic classical bioinformatics. I think very often what happens is bioinformatics. Again, we use the analogy of the parable of the six blind men and the elephant, and that is the flavor of the month is bioinformatics, right? Because somebody says, "Oh, this is the new thing. This is how we define bioinformatics," and that then becomes part of the deal, and which is, in my opinion, unfortunate. But again, because you're studying bioinformatics at a graduate level, depending on whether or not you've had any prior experience with bioinformatics, we have to go through the classical bioinformatics as foundation for the stuff that we do now that is close to the state of the art, and we'll be looking at that, which is bioinformatics applied at the genomics level, and that's what we're going to look at. Okay, so what I will do is my screen will not support every window open, okay, but I have just the top. I just have the top of uh, the Teams window open, and I think that is the so if you've made a comment, if you've typed in something, if you want to draw my attention to something, then I have the top of the mic, the Teams open, so we will be able to see that raised hand. Okay, that is one. And if, on the other hand, I suspect that an hour from now will be shut down. So just immediately, please log back in. Okay. So right now, everybody who's supposed to be in this class is in this class. Okay. So we're looking at sequence analysis, and let's keep going. So before we do the sequence analysis, the foundations of the analysis is the DNA structure. Okay, and the DNA structure is something we have to look at because again, you look for things like sequence comparisons and multiple sequence alignments, etc. But you have to actually understand what the structure of DNA looks like. Okay, now all of you are graduate students with some familiarity with the biological sciences, presumably. So I'm not going to pretend that you have not seen a picture of this kind. But okay, so just bear with me, indulge me. Let me go through some of this because this is foundational, and I think it's a good place to start. So sequence, the structure of DNA. You can see here this is what is called the minor groove, and the minor groove is opposite to a major groove, okay, opposite to a major groove. And what we'll do is once we come to structural proteomics, that is it's essentially the last lecture in this course. Once we come to that, we will actually revisit some of this because here we are looking at the DNA from the perspective of a sequence. There we will look at DNA from the perspective of its structure. But you still need to understand foundationally what this is. And we'll look at various views of the DNA. So you see here is the minor groove, and then opposite the minor groove is a major groove, and then this is a major groove. As you can see, it's kind of this little angle looking thing, though it's not. You have to think in terms of three dimensions. And so this is a major groove, and opposite the major groove is, so when you look at the double helix structure of DNA, it's alternating major and minor grooves. Okay, so now let's look at the color, which is very typical of the color schemes for atoms. And you will have hydrogen and carbon have the same color. It's this white gray, and the only difference is that hydrogen is smaller than carbon, and there is some sort of a height, the size, kind of radius, and so nitrogens will typically be blue, oxygens will typically be red, and the phosphorus is yellow. Now, very often, if you have in your compound a phosphorus and a chlorine, then you have to kind of go in there and look at the different shades, right? And so we all understand. Uh, and so this is kind of an atomic level structure, okay? And so we have the DNA, you have the polypeptide chain, but we have the nucleotides, okay? The nucleotides are adenine, guanine, and we look at this in structure in greater detail. And so you have adenine, guanine, thymine, and cytosine, and then the adenine and the thymine are complementary. And you see what I'm pointing at right now, and I'll show you this, and I'll share these slides with you at the end of the class along with the video. And then you'll see that there are the adenine and the thymine. So you have adenine, which is a purine. It is complementary, exclusively complementary with a thymine, which is a pyrimidine. Okay, and a purine is identified by a single ring. A pyrimidine has two rings, and then you have the guanine, which is a purine, and it is complementary with a cytosine. Okay, and so you have the adenine and the thymine are connected by two hydrogen bonds, and we'll come to what those are later in case you don't know or have forgotten. And then you have the cytosine and the guanine connected by three, right? Connected by three hydrogen bonds. Okay, so that was the previous was an atomic level structure. If you wanted to kind of get a different view, then you have to look at it from the perspective of, kind of shed the atoms and kind of have a bigger bird's eye view, as it were, in order to interact with, in order to look at these interactions, right? So you have the double helix shown by this light blue, right? Light blue, and the kind of the coils, and then you have, this is one helix, this is the other helix, and then you have adenine, which is in yellow, and it's complementary with the green thymine, and then you have the orange cytosine complementary with the red guanine. Kind of a little bit of an easier view, which actually takes away from the atomic level and just identifies these as complementary sticks. And the complementarity is shown here, where you say, okay, well, this is a kind of a wedge-shaped groove, and on the other hand, there is a arrow thing which fits nicely into the groove, right? Similarly, here you have a concavity at the end, right? You can see a concavity which is of the guanine, which is complemented or matched by the convexity. Sorry, the convexity of the guanine is matched by the concavity of the cytosine. Okay, so it just shows you in an illustrative way, in a stylized way, how, for example, this wedge shaped thing would never fit with something which is circular. Okay, that's basically just something that makes it easier to understand. Now, if you dive deeper into the structure of the DNA, you'll see that this is called a phosphorus, a kind of a phosphodiester chain, which forms the backbone of the DNA. So we'll come back to that a little bit later. But here's how you see that I had initially said, so purines are two rings, pyrimidines is a single ring, and you see the two hydrogen bonds which connect them. And similarly, you have the cytosine, the guanine, which is a purine, which is a two-ring structure. And so I flipped them in the previous slide. Okay, and so you have the cytosine, which is the pyrimidine, and you have the cytosine and the guanine. Given the nature of the atomicity of the ring, the atomic nature of the rings, you will have um, three hydrogen bonds. Okay, which tends to make that a little stronger. Now, if you look, coming back to the ribose sugar, ribose is five-membered ring, right? That's the oxygen, five-membered ring, the ribose sugar ring. And the numbering takes place in such a way that the oxygen, which is a functional group of the ribose sugar, it gets priority naming. So it's 1, 2, 3, 4. And then this methylene group, this methylene group is 5. Methylene group is 5. And then you have the phosphodiester. Now, let's go back into the definition of what an ester is. An ester is when an alcohol interacts with an acid, right? an alcohol sorry, my daughter's here watching something. An alcohol interacts with an acid to give you an ester. Now there is always that this is obviously an acid interacts with an alcohol to give you an ester, and you lose a water molecule, and you can look up basic esterification. Now let's ask ourselves this very simple question, right? So typically in an ester, for example, methylformate, right? In methylformate or something acetate, you will have the base, the acetate. It means it arises from acetic acid, and then there is a that the other group, for example, methyl will arise from methyl alcohol. You abstract out of water, and you get an ester. In this case, we call it a phosphodiester. So let's focus on the phospho. That is because the base acid, or the base acid is a contradiction in terms. It's a tautology, if you will, but that's not what we are worried about. The foundational acid is not a carboxylic acid, it's phosphoric acid. Phosphoric acid has a formula H3PO4. Only one of the H's is retained, and it loses two of the H's. So then you have to ask yourself, uh, it's called a diester because here it forms one bond, and another bond. Okay, so that's the loss of water molecules on two separate fronts here. Okay, so phosphodiester, and that's the backbone of the DNA. Okay, so remember phosphoric acid and a diester because there's the replacement of two hydrogen atoms. Okay, of the phosphoric acid. Now, what else do I have to say about this? Oh yeah, then you basically think in terms of you have the 5 prime end because this is the 5 prime carbon, and then this is the 3 prime. This is the 3 prime end. This is ends at the 3 prime. Okay, because this is atom number 1, 2, and 3, 4, and here in this methylene group, okay, the CH2 group is a methylene group. It is 5, so that's the 5 prime end. That's basically what it's called. Complementary to it, you have the 3 prime end ending in the 5 prime end. Okay, so it gives you a bit of a structural perspective of what you typically just take for granted, right? Because after this, what's going to happen is you're only going to represent a DNA uh, molecule by its primary structure, which is A, C, D, G, and you're just going to string them in a line. But I guess it's nice to have a kind of a bit of a foundational as when you just write A, C, T, and G, what really is involved. I'm sorry about this. Hello.
Hi, Chiquito. This is Sharda.
Yes, hi Sharda.
How are you?
I am good. I'm in the middle of class, but I'll send you an email. I think the 10th,
Oh, okay.
10, no, no, 10:30 on Friday is fine. Okay, I'll send you an
Okay,
email to confirm.
10:30.
Yes.
Perfect. Yeah,
Okay?
please send me
Okay,
email.
all
Thank
right.
you.
Thank you.
Bye.
Bye. Sorry about that once again. All right. So now let's go back to the structure of DNA, which is in one sense not really the structure of DNA, but the at least the foundational structure, at least the primary structure is the difference between a DNA. And so we look at the sugar backbone, okay, the ribose, the phosphodiester bond, and then you have the sugar backbone, and that is, you'll see the nomenclature here, and you'll see the difference between DNA and RNA, in that the ribose sugar, at the level of the ribose sugar, obviously there are other things. The thymine is replaced by uracil, which is structurally very slightly different. And you'll see that the oxygen is missing here, so it's deoxy. It's missing an oxygen, deoxy, and then that oxygen is actually present on the 2' carbon atom of the sugar ring. Okay, so a lot of things. This is probably repetitive for most of you, but I think it's a good idea that if you're going to do classical bioinformatics, begin with that foundation. Okay, begin with that foundation. All right, so now we can look at the purines, where you have a fused five-member ring, and those are adenine and guanine, and then you have the pyrimidines, and the pyrimidines are cytosine and thymine in DNA, and the thymine is replaced by uracil in the case of an RNA. So let's look at these structures. So you have a six-membered ring with a five-membered ring, and the best way to look at it is kind of, and they're all aromatic. Or you can see these double bonds, and you can, the aromaticity uh, can be shown in different ways. But here's a difference between guanine, one foundational difference. This five-membered ring is the same. Five-membered ring is the same. Here there is a carbonyl carbon atom in guanine, and this is one of the reasons why you have three hydrogen bonds as opposed to with guanine and cytosine as opposed to one hydrogen bond, right? This is an amino group, an amino site functional group. So that is replaced by a carbonyl group and an amino group. And then here in guanine, the amino group is on this position, and here you have nothing. The rest of the molecule, here there is an NH group, obviously to make sure that the valency is maintained, right? That carbon is attached to 4, 1, 2, 3, 4. Oops, 1, 2, 3, 4, and then 1, 2, 3, and therefore there's an NH group. Okay, so again, nobody is going to, in a test, uh, say, okay, now, close your books and then start sketching what a adenine versus a guanine is. But it's good to kind of, at least because we are studying this, begin to understand how the differences are. Okay, so the thymidine, the pyrimidines, thymine, cytosine, and uracil, are a single ring. Okay, so the single ring, if you look at thymine and cytosine, the same, except in thymine there is a carbonyl group and an amino group in cytosine, and then there's an additional methyl group, kind of like, that's an additional methyl group here, so that's the foundational difference. And then in uracil, so how does thymine differ from uracil? Mostly the same, except there is an H group here. There's just a hydrogen atom, and here there is the hydrogen atom is replaced by a methyl group. So largely the same, except for the methyl group, which gives thymine a bit of a greater bulk. Okay, now let's begin to understand that the only reason, and you really don't think of a hydrogen bond as an actual bond in terms of exchange of electrons, but if you think about the sustenance of the DNA double helix, they are not in any way connected in a kind of a covalent bonded situation, but they are bonded by hydrogen bond, because you take away the hydrogen bonds and you do not have the double helicity of DNA. It basically splits into two single helices, right? And so let's ask ourselves, how do adenine and thymine organize themselves, right? And then later, guanine and cytosine organize themselves in such a way that the hydrogen bonds are are placed here, hydrogen bonds, and then three hydrogen bonds here, right? So you see, this is thymine, this is the NH2 group of adenine, which forms a hydrogen bond with the carbonyl group here, right? Carbonyl group, and then here there is a relatively weaker, because that hydrogen bond. And then on the other hand, in terms of guanine and cytosine, you see that here's how the hydrogen bonds. This one, the middle one is similar to the middle one is similar to this one here, but you have two of these carbonyl group, amino group, carbonyl group, amino group, hydrogen bonds. Okay, I'm looking at no chat, no hands raised, that means everything here is good. Okay, now, so if are there any questions? Once again, uh, I cannot have all windows open and in full view. I have them open, but I do not have them in full view, but I can see the top of the Teams, so I can see the chat, and this class has been, I think, going on for 44 minutes or so. So uh, we'll see. Okay. So again, if we are suddenly cut off, just log back in, and we'll continue where we left off. Okay. All right. If there are no questions, I think the question then begs itself: for those of you who forgot, or those of you who don't know, or never remember, or remember it from a long time ago, what is a hydrogen bond? Now, here's what I'm going to do. Flip over to, I'm going to flip over to the uh, Teams, and if somebody wants to, again, you don't have to have the right answer, but if somebody wants to provide me with a an opinion, somebody wants to provide me with an opinion of what they think a hydrogen bond is, I'm all ears. Anybody wants to define a hydrogen bond for me? You can just mute and tell me. I can almost hear the theme music to Final Jeopardy. Maybe I should play it. The theme music to Final Jeopardy.
I can get us started maybe, but
Yeah, go
I
for
don't
it.
quite remember. It's been many years,
All
but
right, go for it.
I know it's not the strongest bond. Like it's easier to undo and then put back together. Very so it's kind of easier like that. And a hydrogen is I think it's not it's more of like with the ion charge kind of attracted to the opposite charge with the oxygen maybe could be more negatively charged and but it's not like a really strong bond.
Okay, I think you've gotten us started correctly. Okay, so thank you, Michaela. Anybody else wants to elaborate to qualify what she said? Okay, I can almost hear crickets. All right, let us now begin to look at a definition of a hydrogen bond. Now, there's some other questions that we have to ask, right? For example, what are valencies? That is, what is the bondability of something? So, for example, if you see that the valency of nitrogen, okay, is 3 or -3, which means it has to be associated with 3 bonds. 1, 2, that's from the double bond, and 3. Carbon is 4, so 1, 2, 3, and 4. These two come from the double bond. Oxygen is 1 and 2. Okay. There's another question I asked is hybridization. And again, I'm not going to elaborate a lot of this because a lot of this is kind of foundational undergraduate work where you talk about hydrogen. For example, hybridization. Why does carbon form? Why is methyl group has a certain geometry, and that comes from hybridization. And so you think in terms of sp2, just to kind of jog your memory, sp2 hybridization, sp3 hybridization, sp2 is where you eventually end up with a double bond, sp3 hybridization, and then you go when you go into the d orbitals, you begin to look at metals, which is again the foundations of the organometallic chemistry in the world. And then you begin to look at sp3 d2 d2 sp3. There's a whole bunch of hybridization. That is the orbitals. And what is an orbital? Orbital is, like, let's look at it from the perspective of quantum chemistry, is the probability density within an orbit, the probability density of finding an electron. Okay, the probability. So if you say, an s orbital has a spherical shape, does not mean that the orbital has a spherical shape. The orbital is where it is most likely to find that electron, and each orbital has 2 electrons, so is in a sphere. Now, if you remember, the uh, the the p orbital is a dumbbell shape, and the dumbbell shape doesn't mean that the orbital actually has a physical dumbbell shape. It means that the electron has that probability of being in that figure of 8 kind of a thing. And then the d orbital gets more complex because the dz orbital gets more complex, but you have it has a double dumbbell shape. Okay, that's where you're likely to find the electron. So when you have all these orbitals, basically there is a way in order to you can get together and form a hybrid orbital which has features of one and two. And the easiest way to explain that is in methane, right? Methane, the formula is CH4. And I'm just talking about these things. You guys can take notes and, again, look for it. Is methane is CH4, and if you try to organize CH4 from its structural standpoint, you realize that all the hydrogens are exactly equivalent. Okay, so how do you just add them, attach them to the carbon in such a way that they are all equivalent? You could make them planar and make them at kind of corners, but they found out that they have a uh, the has a tetrahedral geometry. Okay, and once again, I'm saying these things. You can go look this up. For a lot of you, this is a refresher. So these are tetrahedral geometries, and the hydrogens have only s orbitals, right, which is only one electron. The carbon has an s orbital for its, and then three p orbitals, right? And so what happens is that the connections of the methane has basically just four identical orbitals, which is why all the hydrogens are completely equivalent, and that is because the s orbitals of the hydrogens and the p orbitals of the carbon have gotten together. The 3p and 1s has gotten together to form four sp3 hybridized orbitals. So that 1s and 3p's. So each of those bonds is an sp3, and therefore four of those have been created. Okay, so that is where you have a hybrid. You have two completely different orbitals getting together to form a new type of orbital that has features of each of these orbitals, and that's hybridization. Now let us see what a hydrogen bond is, and I think Michaela, you alluded to some of this. So let's say, for example, if you look at the periodic table, you will see that anything as you go down this list, so this is more electropositive. On this side, we have more let's not worry about the hydrogen and the helium because the chemistries are different. But let's say from here, from lithium, you'll have more electropositive on this side, and the electron positivity decreases and the electronegativity increases as you go from here to here. Also, the electropositivity increases as you go down the group. So this vertical is group, and horizontal is a period. So you go from this side to the other side of a period, and the electronegativity increases, the electropositivity decreases. As you go up a group, the electronegativity increases, the electropositivity decreases. So if you go down a group, the electropositivity increases, and that means as you go down a group, the electronegativity decreases. Okay, so fluorine is the most electronegative of the compounds, right? And when we go back to the discussion on valencies, you see that fluorine just requires to interact with one other atom. Oxygen requires two, nitrogen requires three, and carbon requires four. Now, carbon, based on electropositivity and electronegativity, is neutral and stable because it can kind of accept, reduce, or give away four, but the energy to accept four electrons to form kind of an electrostatic bond or an ionic bond is significantly higher, right? And therefore carbon, basically all of organic chemistry, all the chemistry of life is organic based, and that is based mostly on covalent bonds. Okay, so that's kind of the point of it. Now, if you look at something that's more electronegative, right? When you have something that's more electronegative, you will see that you will see that when you have an atom, for example, this is ammonia, NH3, this is water, H2O, and hydrogen fluoride. Because this nitrogen tends to be more electronegative, the oxygen tends to be more electronegative, as I showed you, these are the more electronegative compounds. You will see that in a covalent bond, where you say that the electron, not the electrons, now we're graduate students, so we say where the electron densities are shared, they're not really shared, right? So that while the electron densities are shared between these bonds, this nitrogen will, because it's more electronegative, will drag away that entire electron cloud towards itself, right? And because it's dragged it towards itself, it is more tends to be has a what we call not a full negative charge by a dash, a negative sign, but a delta negative charge. Okay, it has a delta negative charge. Now the hydrogen atoms, on the other hand, because they have lost that one electron which they're supposed to have, which is supposed to share, they will then gain a slight positive charge. Okay, slight positive charge. Now it's the same thing with the hydrogen fluoride. Right, hydrogen fluoride. Those of you who watched Breaking Bad are very familiar with the reactivity of hydrogen fluoride. And so, because fluorine is electronegative, it dragged a lot of the shared electron density towards itself, making it slightly negative, right? And here's each of these red circles, red balloons, so to speak, are represented by electron pairs, and therefore this hydrogen now is more positive, has a delta positive charge, or kind of a partial positive charge, because all this is given here. And the same thing with water, and water here is very crucial. Okay, so what happens then is, when this water is associated with another water, right? Let's say there's another water here, and I'm going to, there's no way for me to actually, I suppose I could draw it, but let's say there's another water here, there is another oxygen atom here, and two hydrogen atoms, and it also has an electronegative charge here. You will see that water will form these very, relatively speaking, weak bonds. As Michaela said, you could easily break and easily form with four of these, right? So, and that bond, not this bond, this is the proper covalent bond, just with a kind of an unequal impartial distribution of charges. On the other hand, this one, the electrons that it'll form with this oxygen here that I'm drawing, kind of fictitiously, I'm drawing like a ghost oxygen. You'll see that that is a hydrogen, that is called a hydrogen bond. So, and this is really important, because a couple of years, for 2 years, I used to help with our Center for Biotechnology and Genomics, MS in Biotechnology, and whenever we had candidates who applied to our program, after, if they met the first requirement that we said, that they met the metrics of our program, and it's like, okay, we can accept them. We actually did either a face-to-face or a Zoom, etc., interview with them, and we found that I used to ask them nothing fancy, all the great things that they purportedly have done in their based on their resumes and their transcripts. I used to ask them a very simple question, and that is, can you tell me what a hydrogen bond is? Okay, and it was very rare that anybody knew what a hydrogen bond is. Okay, but at least from a biochemistry standpoint, as you become foundational biotechnologists, begin to understand that nothing happens without the hydrogen bond. Okay, it's the as Michaela said, it's not a real bond. It's kind of a distribution of charges because of the nature of the chemistry of the compound involved. It is very crucial, because there would be no double helix without the hydrogen bond. Okay, and once we come to a greater discussion of the structure of an alpha helix or a structure of a beta sheet, which are the foundations of secondary structure for proteins, once again, without a hydrogen bond, you have no secondary structure. Without a secondary structure, you have no tertiary structure. Without tertiary structure, you do not have that unique notion of protein folding, and that is one of the challenges, so that is why it's a small bond. It wouldn't even qualify as a real bond. If you put it in some sort of a thing, there wouldn't be any hyperfine structure that a spectroscopist would typically see, but you will see that they are very, very crucial. Now, the question then you can ask yourself is, in terms of how much does the hydrogen contribute, and why is water the solvent for life, right? Why does nature or whatever create water in such a way that it is just fluid where we need it to survive. Okay, which means, and a large portion of our bodies are composed of water. And why is water so crucial for life? Okay, and so that can be seen from by understanding why you have now these are the boiling points, boiling points, boiling points, boiling points, right? And I'm not going to worry too much about, as you go down, it's oxygen, sulfur, selenium, tellurium, okay, or fluorine, chlorine. Those are easy. Bromine, iodine, those are the halogens, and then ammonia, phosphorus, arsenic. SB is tin, and Bi is bismuth. I'm not worried about that. I'm just worried about the same thing. If you take the hydrogen atoms, from the hydrogen bond perspective, you will see that water has a boiling point of 100 degrees Celsius, that you know, and hydrogen fluoride has a boiling point of 20 degrees, of, right? And then ammonia, which is ambiently and otherwise mostly a gas, because it has a boiling point of negative negative, 30 something uh, Celsius. Okay, so the question you have to ask yourself is, why do we see this anomalous behavior? So if I go back here, I told you that fluorine is the most electronegative, then oxygen, then nitrogen, right? And here you will see that that means that because this distribution of charge is the highest, because fluorine is the most, right? Fluorine is the most electronegative. This should be the most electropositive. So if another fluorine shows up, HF shows up here, that that bond, hydrogen bond, should be the most strongest, right? Most strongest means harder to break, right? The strength of the bond is always defined quantitatively by the effort it takes to, the energy it takes to break the bond, right? And so that the hydrogen bond should be the strongest, right? And yet, if you really see, that has 20 degrees Celsius is where hydrogen fluoride. So at kind of very cold temperatures, it's a liquid, right? And so then you have, in the case of a, so that is, so why is water, which oxygen is not as electronegative as fluorine, so the hydrogen bonds associated with them are not going to be as, they're not going to be as strong as the hydrogen bond associated with hydrogen fluoride, yet it has a boiling point that's higher. Okay, and then if you have ammonia, which is 3, right? So it's this interesting, so to speak, anomaly, easily explained. But I basically am now going to open the field for you guys to explain to me, why do you see that anomalous behavior, though it would be the hardest to break a hydrogen bond associated with hydrogen fluoride, okay? And but however, water has a significantly higher boiling point. And on the other hand, if you say, well, maybe because it has more bonds, more hydrogen bonds, right? Because H2O, then you would say, then why does ammonia have such a low boiling point? And now I'm looking for opinions. Anybody? I really need to get that. I need to get the music from from Jeopardy, just to make these pauses more dramatic. Okay, the meeting will end in 15 minutes. All right, so we're good. Let's not worry too much about 15. And as we come to 15 minutes, I'll just leave and then we can rejoin. And again, I should have done my due diligence, uh, and asked myself why they won't have. But once again, you have the links. Come right back in, as will I. So, anybody has an opinion? Again, I'm not saying that your I'm not saying that your answer should be correct. Um I'm not saying your answer should be correct. I'm just hoping for an opinion. Okay, so the answer actually lies in the so while hydrogen fluoride is the bonds as hydrogen bond associated with hydrogen fluoride would be the strongest. Every hydrogen fluoride will only form one more, right? So there's with one hydrogen fluoride and a surrounding hydrogen fluoride, there's two hydrogen bonds. With oxygen, right, there are four hydrogen bonds associated with one molecule. It's attached to four different. As oxygen is attached to two, and then each of its hydrogens are attached to two. Similarly, in the case of ammonia, there are six. But what happens here is you have to ask yourself the relative electronegativities, right? Hydrogen fluoride is very strong. Oxygen is somewhat less strong, but in ammonia, the nitrogen electronegativity is actually quite weak. So though it forms two, it forms more hydrogen bonds. Its electronegativity is not strong enough to have strong bonds. So the bonds are really weak and can be easily broken even at really, really low temperatures, right? Approaching negative 40 Celsius. Hydrogen fluoride is stronger, but there are not enough bonds. And so we have this really, with water, we have this really happy medium, okay, this really happy medium which allows it to be the solvent of life. Okay, so again, there's a lot of kind of interesting chemistry which goes towards saying that, why are we composed of water and not another solvent? Water is a universal solvent. Yes, it cannot dissolve anything, but it can dissolve a lot of things, right? Um, Again, we can drink water, right? We can drink water, we can bathe in it, but because somehow, and this is not the only reason, I'm just saying that somehow the combination of hydrogen bonds contributing towards this behavior of water makes sure that it is fluid and accessible at temperatures which are which are typical of the Earth's temperatures and the temperatures in which we can survive. Okay. All right. Did anybody say anything in chat? Adhesion, cohesion properties the same. So, okay. So I didn't even check the chat. But again, like I said, if you're going to type something in chat, because I can't see just raise your hand because there's a little red dot which comes over chat which I may or may not see. So adhesion, cohesion properties the same for so you have to ask yourself what is cohesion and what is adhesion. They not have much to do with the specific definition, but let's address it. So Maylene, obviously I've given you your answer. So I don't think it's size as opposed to a combination of the optimum electronegativity combined with the number of hydrogen bonds. Okay, so that's the answer. Uh, but adhesion. So like what is cohesion, right? Cohesion is where like will interact with each other. Adhesion is where unlike. Now typically we say, oh, adhesive is a glue, right? Glue is a glue stick is a glue is an adhesive, or a whatever compounds they use now to create the super bonding agents is. So the adhesion cohesion properties is same for each of these. Yes, they are, but it's the underlying chemistry which which defines this. Okay, so again, sorry about that. I missed the chat, but again, if I see a little raised hand thing, then I'll realize that you're trying to get my attention, and then I'll look at chat. Okay, as I said, the screen is not big enough for me to have everything open here. Okay, so that gave us a sense of the foundations of the structure of DNA, and I guarantee we will revisit this at a later towards the end of the class, because then we'll actually focus purely on structure. So now there's some philosophical points that are going to have to be discussed. What is sequence alignment? So let's look at the basic definition. A sequence alignment is a way of arranging the primary structure. Now, when you think of primary structure of a DNA, would be just literally writing the nucleotides in a straight line from 5 prime to 3 prime, and just identifying that from left to right is left is 5 prime, right is 3 prime. Right now, you're kind of seeing it the other way around in the camera, but that's fine. And also, a primary structure for protein is basically writing the entire sequence of amino acids of the protein from the N terminus to the C terminus in a straight line. Okay, we're not concerned about the structure. We're not concerned about the atomicity. It's just basically just writing it. Now, the question you have to ask yourself is, why do we need to align sequences? Okay, all right. So see, because if I have a sequence, if I have a um, DNA's, whose sequence I know, by aligning it with other sequence, I can say, hey, if this sequence aligns well with a bunch of other sequences, it's possible that they have the same structure, right? If they look the same, they likely do the same thing. And then you can also say that in addition to that, I can also see how things worked out in evolution. If two things that look dissimilar have certain things that are the same, you could say with some slight difference, you'd say, hey, maybe something evolved from something else, right? And then you have to say that when you align two sequences, the second point is it about similarities? Is it about identity, right? Identity means identical. Similar means same, right? So, and this is more obviously seen in the case of amino acids, right? So, for example, and again, I don't know how much you know of amino acids, but just for example, you have an amino acid that is basic, it's alkaline in nature, and you say, oh, okay, if a lysine is replaced by an arginine in the other similar structure, you would say they're both positive. One is slightly bigger than the other. Uh, Positive in the sense they are both basic. Not much is going to change in terms of its function. Something small is going to change in the structure, right? If on the other hand you have the glycine, which is just an H, is a side chain of an amino acid. Glycine is just an H, but it's replaced by phenylalanine. Phenylalanine is where you have a benzene ring as a side chain. And it's like, this is very curious, right? It's not similar at all, which means something different is happening. Whereas if a glycine is replaced by an alanine, where the H is replaced by a CH3, yes, it adds a little bit of a bulk to the side chain, right? So that's what you want to see. And then to recognize orthologs, homologs, and paralogs. Now, orthologs, everything is a homolog. Homologous sequences very often are interchangeably used, but very often incorrectly used. Oh, those two sequences are homologous. Not necessarily. When you say something is homologous, we say that in a kind of a hand wavy way, we say that they're homologous because they are similar, or they're homologous because they're identical. But that's not the real definition of homology. A few slides from now, we'll actually look at what the definition of homology is. On the other hand, what is an ortholog? Ortholog is where sequences that have that arise from that have the same function but arise from different species. And you would say, that's a mouse sequence, and this came from a human, but they're very close to each other. Is it possible then that particular sequence, whether it's DNA or then the translated or the transcribed translated protein, do they contribute to the same structure? They are similar, but they come from different species. Or then alternatively, you might have something called as paralogs, which are sequences that don't look the same or look the same, but they come from the same species. Okay, or sometimes they might have just different functions because they look different or they're slightly different from each other. So the question is, why do you need sequence alignment? There is a philosophical answer to this, and the philosophical answer is, well, we'll get to it. Now it looks like we're going to get kicked out anyway, so I'm going to I was kind of going to the next part of the discussion, but I do not want to be rudely interrupted. So I'm going to, okay, so here's what we're going to do. We're going to leave. You have the link. Leave, and we're going to come right back to the same link, okay? I have to do it one more time. And again, I'll try and be a little more diligent and ask for just a full 3 hours so we don't have to do this all the time. I used to use Zoom, and it did the exact same thing with Zoom. So I'm going to leave, come back, so this way we can have an uninterrupted conversation. Okay, everybody's back. Thank you very much for coming back. Uh, Let us go to sequence alignment. Now, before we go into the kind of the philosophical discussion as to why we need single, sequence alignment, sequence similarities, etc. Let's look at a kind of a real life, which part of my personal research coincides with this. Okay, so I don't know whether you know this, that as soon as the human genome, the first version, the first edition of the human genome came out in 2000, 2001, etc., a lot of people decided that we want to mine from the human genome olfactory receptors. Now, about 10 years ago, people started doing research on olfactory receptors. So what are olfactory receptors? Let's go here, and we will revisit this database again. So I just want you to explain, because I'm actually the curator of this. I continue to be the curator of this database. So an olfactory receptor, we believe, looks kind of like this. Now there is an actual X-ray, not X-ray, but there's a cryo-electron microscopy which came out 2 years ago, but it kind of looks like this. So an olfactory receptor has 7 helices. Okay, they are a transmembrane, so they are in the membrane where the one part of it is outside, open to the atmosphere, to the environment, and the lower part of it is cytoplasmic. Okay, and this forms like a barrel-like shape. Now, what they found was that olfactory receptors are the largest family of proteins or genes in the human genome. Okay, it's the largest in the human genome, literally 20% of the genome. Okay, now four separate groups, research groups, over time, over 4 years, 5 years, they went and said, typically in a gene, now you can look at associations and all of that, that several genes, that, okay, sometimes you might have one or two genes responsible for a particular phenotype, particular function or a particular characteristic. But in this case, you had 400 functional genes, okay, approximately, give or take, and they actually found that there were 1,000 genes. So I guess it does not allow me to kind of recreate a meeting until we've been all kicked out from that meeting. So I'm sorry, and I'll try. If somebody can send me a reminder sometime in the middle of the week to just deal with this, I would appreciate it. Okay, and Chuangwen, please, can you mute unless you want to say something? The Thank you. So again, we are looking at a. So what happened was different groups went and they mined from the human genome what they thought using sequence similarities to each other. 10 years ago, there was a very interesting the, the first olfactory receptor was identified. It was a single thing. It was sequenced, and they knew what it looked like. And then they found them all, began to find them all over the place, and they found out there are functional genes. Out of approximately 20,000. If you go to gene, depending upon all the variations and all, there was about now you'll find probably 60, 70,000 variants. If you go to the human genome, um, the Human Genome Organization, the consortium, you'll actually find there's like 95,000 genes. Most of them are variants and things like that. Okay, so 20,000 genes. They found 400, and they said originally there were 1,000. But so these looked same, based on sequence similarities, sequence identities, and things like that. Except these were the 1,000. About 600 were pseudogenic, non-functional, and various reasons why something could be a pseudogene. You could have a sequence that has just a stop codon, a nonsense mutation, what is called a nonsense mutation, right in the middle of the gene, which renders it non-functional. And so there were all these things. So they did a pretty thorough analysis, and four separate groups found out, and they found out that about 300, somewhere between 375 to 400 were functional. And the four groups did this independently, and that doesn't mean that anybody agrees on anything. Okay, so the reason why this is a good illustrative example of sequence similarity is because the question was, how does one organize such a large family of genes? Okay, how does one organize a large family of genes? And so one group said, let's basically do a sequence similarity of the entire group of genes that we have, and if they are within 40% similar to each other, we will call them a family, and then just cluster them on families based on 40% similarity. And then they said that within a family, let's do a further clustering, and if they are 60% similar, we will call them a subfamily. Now, this is not Moses descending from Mount Ararat with the Ten Commandments saying that this is what is supposed to be done, right? There is no hard and fast rule which said that those two belong to a family, those two do not belong to the family, and then those two belong to a family, but they belong to different subfamilies. I think I hope I'm kind of getting to that philosophical point of sequence similarities, why we do them, why we don't do them, why they are necessary. Okay, and that is this this number, right? This number was arbitrarily determined as 40%. This number was arbitrarily determined at 60%. Because the entire organization of the genome would have been completely different if they said, well, if it's 38% or 35% or 30%, we'll call it a family. Then the organization, the clustering would be completely different. You would have more groups, more genes in a group, because they would not be the same, right? Then you would see then, and again, you could just say that within a group, if it's 50% similar, we will call it a belong to a particular subfamily. Okay, that is one group that did that. then there were several other groups that some people just was like, and then this particular family subfamily situation had nothing to do with chromosomal location, for example. Then there were others who actually said, so let's go back to this group, said that if this belongs to a particular family, it'll be numbered. Okay, like one, family two, family three, family four, etc. And then within that subfamily, it would be A, B, C. So, for example, if something belonged to family 3 and subgroup G, it would be ORG. And I'll show you the actual nomenclature system. That was one group. The other group decided that we don't even care about this. We are going to basically going to number them in such a way that it would be based on chromosomal location. Okay, and it was chromosomal location. Within the chromosome there was a family, and then within that family they just wrote the numbers in ascending and descend in just in this kind of a numerical way. It had no real meaning other than just a number was assigned to it. Then some other people did something else. One group did nothing in terms of chromosomal location or anything. They just lined up everything, and based on how they identify them, they just numbered them. Okay, and so again, there's kind of a combination of a science and an art, and you can kind of make a point that this is my reasoning as to why something has to be done. Now, the group that was able to do this family subfamily, they actually convinced the Hugo, the Human Genome Organization people, because they said, well, now we have the human genome, now we have to begin to organize something. They convinced them that this should be the accepted nomenclature. I'm not going into the politics of it because I don't know exactly how that happened, but they said that that was going to be the nomenclature and not the others. And I'm going to give you an illustration of everything else too. So the Hugo people came to me. The reason they came to me was because at that time I was I still remain the curator of the olfactory receptor database. Okay, you see right now, if you look at the address here, it's not very, maybe not very clear. It says ordb.biotech. So we took that entire database and we kept it at the Center for Biotechnology Genomics, where, this is the where you're getting this course from, and we called it ORDB. Now, what the Hugo people said was, we have decided to adopt that methodology, that 40%, 60% similarity, that nomenclature as our standard for olfactory receptors. Then they made one big mistake. They came and told me, you should do the same. Now, there is one primary issue with somebody telling me what I should and shouldn't do. It's not going to happen. Okay? I think the last group of people who told me what to do and not do were my parents. After that, it stopped. Um, but again, we still are very interested in the scientific rigor. So what I said I'm going to do was, I said I will do something a little different. I cannot, because again, this was, and I was not even a faculty. I was actually a postdoctoral fellow at Yale University at the School of Medicine, and this is where this database was housed, and I was the primary developer with a colleague, but I was also the curator of this resource. And I said, let me do this, and I'll show you this web page. Okay, so you see, this is the methodology which I said. They said if it's 40% similar, like this is the first on the first chromosomal location and the first. This is I gave it a consensus. But I said, this is the consensus, but honestly, I don't care if you adopt it or not. It doesn't matter. Okay, it doesn't matter because they say that nomenclatures, the scientists would rather share toothbrushes. This is something I've heard, than each other's nomenclature, because everybody is really adamant. They're not going to shift. This is what we name it. This is what we named it. Okay. And so this is the group, and this group was at the Weizmann Institute of Science in Rehovot in Israel, and they said that So you will see that this is an olfactory receptor, OR. This is the family. It belongs to family 10, and it belongs to subfamily K, and this is the first member. Okay. Now let me make this a little smaller so we can see this a little better. And then you had a group called Genomics, which is a company which did this work, did something else, got dissolved, and now if you type in Genomics, they do something completely different. And I suspect it was sold to somebody else, and certainly that one individual who worked with me, he's not there. So what did they do? Well, they said that we are going to worry about chromosomal location, and this is a family within the chromosomal location, and this is just the number. And then you had the European Bioinformatics Institute, and his name was Michael Croning. And Michael Croning just numbered them, just called them AGPCR. Okay, this was the database that I maintained, and I just also called it the same thing. But we just said, because we had started populating this database of olfactory receptors and taste receptors and things like that way before the genome came about, right? So I was not working there, but the people who worked beside me, they had put stuff based on, just scouring the information, looking at GenBank, looking at this information, and just adding it. So we saw no reason to change it. And then you had the lab of Masatoshi Nei. Masatoshi Nei also this was the most recent of the way in this it was done, and so he calls it OR1, but he focused on chromosomal location. Now, the reason why this becomes, for those of you who are just like, well, we just put something in BLAST, we're going to get similar sequence. The reason I'm illustrating this is because this becomes really crucial, right? Uh, because so what happened, you will see that I said, okay, I'm not going to get into a fight. I'm not going to necessarily favor one group over the other, including the work that we had been doing. And I am going to take all these sequences. I'm going to go to each of the sources and I say, give me your sequence. And I'm going to run my own sequence analysis. Okay, so I wrote some scripts, wrote some code, wrote some programs which actually did this. And I found these similarities, like 100% similarities. But very often, I found that they were not such. So you can see what the color code is. That yes, it was there, but depending on how they didn't mine the entire open reading frame. They mined some of the sequences had partial matches to a full sequence match. Okay, so those are different. Then you have these which were identified again as partial sequences, but identified as pseudogenes, which some groups didn't identify as pseudogenes. You see, so what I have to be is I have to maintain my integrity and preserve the integrity of information that was given to me. Okay, and so things kind of started getting, now yellow is purely pseudogene. This is a partial match with a pseudogene, so I colored it so people can come in, get all the information, and we say this is our consensus sequence. Doesn't mean you have to consent to anything, but if you look at this sequence, you will get this sequence that comes from Genomics, this sequence comes from the ORDB, and this comes from the Masatoshi Nei lab, right? And so, which was at Penn State University. And so you'll see that these are the people who Hugo has adopted, but there are so many sequences that they found that others didn't, or others found that they didn't, right? So it's a big mess. At the same time, this, in my opinion, now if you look at this thing, you see the red areas. The red areas is where the two groups, they did not even agree on the chromosomal location. And sometimes they said this is on chromosome 5 or 5 and 6 or chromosome X. Okay. So I basically organized it. It took me a long time. It took me a lot of effort. And I actually recently, after many years after this fact, I actually wrote a paper which explains this. Okay. And then the other thing that we did was also, then soon after that, the mouse receptors came out. But in that case, we had just two groups, one from the Fred Hutchinson Cancer Research Center, and another from Columbia. And so we did the same thing. And then some, and again, because I maintained this database and as a web resource, sometimes people came to me and said, hey, this is the mouse, but can you include the human orthologs, and that is where we included the human orthologs. Okay, so again, this is a, you might or might not agree, but this is a big deal. Okay, this is a very big deal. But I just wanted to illustrate that yes, it can be a small thing. You say, oh, I'm going to do a binary sequence alignment. I'm just going to find a bunch of things. But at the same time, when something comes at you from the genomic level, then you have to then you also have to kind of have to philosophize the problem, and begin to kind of intellectualize it to a point where you say, okay, I have been cast as a leader of this particular thing, and I need to now make a decision. At the same time, I have to do it in an unbiased way that nobody else, that I do not favor one group as opposed to the other, because each of them has a perfectly solid rationale for what they did, and why they did it and why they gave it. Now, though the Hugo people though the Hugo people considered the one that came from Israel to be the primary one, right? This group, the Horde group, it's called the Human Olfactory Receptor Data Exploratorium. Now, I've got some good friends there. We have worked together, but at the same time, I have to give credit for everybody who did the work. And again, over time, as more versions of the genome became available, right now we have the T2T, telomere to telomere. That's the best one. And I didn't do this work myself, right? My job was to maintain a particular resource in addition to all some of the other research. This was one of the things I did. What I wanted to make sure was that everybody got an equal hand, into this process. Any questions before we go on? No hands raised, no little red dot over the chat, so I guess we're good. Okay. All right. So now let's begin to look at these definitions of what a homolog is, what an ortholog is, and what a paralog is. Okay. Give me a second, please. I'm in the middle of class. You cannot come here. No, she's not coming because she had a tummy ache yesterday night, so now she's sleeping in her bed. No, maybe tomorrow. I'm teaching class, Radhika. What are you doing? Tomorrow she will come. Okay, so as I said earlier, very often that people use homology with sequence similarity, sequence identity, and they do that in a take it to that table. Take it to that table, please. Take it to that table, please. Thank you. And very interchangeably, incorrectly, right? Because they say homology. Now, what exactly is the definition of homology? In order for two sequences to be considered homologous, addition to identity, similarity, etc., they have to arise from that common ancestor. So if you don't arise from a common ancestor, the two genes, no matter how identical they are, cannot be considered as homology. Now, again, going into the philosophical aspect, if you say that everything arose from a common ancestor and just branched and kind of bifurcated and divided, and parents gave rise to daughters and parents gave rise to daughters, um, it has to come from a common ancestral DNA sequence. Okay. Now, a homolog may apply to a relationship by, so how do they get separated? By speciation, right? Over evolution, you get enough of a variation that you might have daughter species, or you might have genetic duplication, right? So speciation is a formation during evolution, a form of a new species. And then look at gene duplication. So for example, you might have a new sequence, so largely similar, right? So here's a sequence, and then this entire red and yellow sequence gets duplicated. And you can take a look at has take a look at what the definition really means. I just wanted to give you a bit of an aside as to what that is. Okay, what is an ortholog? An ortholog are genes from a different species that evolved from a common ancestor. So orthologs also are homologs, but they have the same function. And so, for example, when I showed you this resource for the mouse, right? Let me go back to this. Uh, we saw that yes, this is the group, this is the olfactory receptor, this is the one that I was managing, where we put all that information in, and that is the human ortholog, which means it's an olfactory receptor, but it had really good similarity, right? Though it came from a different species, it was included here. Okay. So what else? Then you have what is called a paralog. So paralogs are related by duplication within a genome. So while orthologs retain the same function, paralogs might evolve new functions. However, the thing about orthologs is they come from different species, paralogs come from the same species. Again, what is speciation? Is the origin of a new species capable of making a living in a new way from the species in which it arose. So some basic foundational definitions. Okay, so now illustrate this a little differently. So again, homolog is that umbrella in the sense that everything arose from the same common ancestor. And then you had, for example, you have the frog, chick, and the mouse, and let's assume that alpha is a function and beta is a function. So a frog, chick, and a mouse, right? The frog, chick, and the mouse have a particular function, but the function is the same, though the species are different. So that would form under the category of. Now, due to whatever reason, if that function evolved into something different, yes, the mouse, chick, and frog beta is an ortholog, right? But the mouse alpha and the beta are paralogs, right? And so you can say, this is the globin gene. And what are globins? For example, hemoglobin comes from that globin gene. Myoglobin comes from the globin gene. So again, you have an alpha chain and a beta chain. And in one sense, you can also think about it this way that, and we'll come to this when we do structure, that the alpha, the hemoglobin has four subunits which are slightly different. Two are alpha and two are beta, whereas the myoglobin, which is responsible for oxygen transport in muscles, is only one subunit. Okay, so that's kind of just a little bit of a context for you, but again, that's how you will determine these. Okay, now because we are looking at this, we are looking at duplication, speciation, and that means the DNA changes. The DNA changes, but in a substantive way, right? Or many DNA changes in substantive ways to ensure that you actually get a new species that functions differently from its parent, right? Or its system. Okay. So the types of mutations, consequences during alignment, if you have an insertion of one more nucleotide base pairs, okay, into a DNA sequence, which then transcribes differently and then consequently translates differently. Alternatively, you can have a deletion, and when an insertion and a deletion happen kind of in a combined way, they're called indels. So here's an example, which from this perspective would be a deletion, whereas from the perspective of sequence 2, it would be an insertion. And then you can look, for example, when you do the sequence alignment, you would see that the ones that are aligned are the same, and then the X's would be for where they are completely different, right? So you can see the X's where they are completely different, and then you would have a single deletion and an insertion identified by an X, and you can kind of move things around, and we'll see what that means. Okay, and that's a gap, and we'll see. So once again, this is the question I want to. And there's a reason why I'll send you this PowerPoint at the end of class, but I do not want to send it at the beginning, is because I want you to kind of, start kind of jogging your processing power to. So the question you have to ask is, um, classical bioinformatics arose from the need to align, right? sequence alignment, binary alignment, blast searches, which we will do, all of this arose from this idea of alignment. Everything is aligned. And I told you, you align because you want to find a similarity somewhere. And that similarity will tell you that an unknown protein or an unknown DNA has the same function because it looks the same, right? It has the same structure because it looks the same. Maybe there's an evolutionary relationship between two things, so that's why we align. So that's kind of the philosophical point. But I wanted to take you one step back, and that's why I asked this question. I asked this question again. Okay. Now the question is, from the perspective of why we align, right? In foundationally everything. And now, if you say, well, that was classical bioinformatics. What about next generation sequencing? Well, it works the exact same way. It's just that you take the stepwise align the aspects of classical alignment and just apply it on a grander scale. Apply it, you take a small sequence and align it, and then you say, I want to actually align something to an entire gene in a known genome whose sequence is published, and then I want to then basically extend it to every gene. Okay, but the foundational thing is still classical, and then is classical bioinformatics or foundational bioinformatics, and we have to understand this. So this is but it's also a bit of a philosophical question. Why we align, how does nature align, but more importantly, does nature even align? And that's a question you have to ask yourself. So I'm going to look for chat opinions. I'm looking for opinions of people telling me what they think. Okay, I see Michaela. I've got the chat open, so I want to see what's going on here. So again, I see whoever's typing is free to type, but on the other hand, if you want to unmute and also just share your opinion with everybody, that's perfectly fine. That is a very specific example, right, Michaela? Like that is a example. That is, you want to defend a bacteria to defend from virus infiltration. Okay, that is a very specific example. But what I was looking for, and thank you for telling me about this. Thank you for sharing this. What I was looking for was why does any alignment take place? Does nature need to align? And you have a little bit of that metagenomics bent to what your answer is. But does nature align? Why does anybody align? So go back to this question. Why do we align? We align because we want to make sense of nature. I think that's in one sense that is the question I was looking for, right? We align because we want to make sense of nature. Let's assume there was an indel, right? What is an indel? Because when nature creates a DNA or and then consequently and subsequently and consequently a protein, does it have a gap, right? Do you ever have in nature nucleotide, 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 nothing, 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 nucleotide, nucleotide, nucleotide? There is no nothing. There isn't, there's no such thing as a gap where there is nothing. In nature, there is nucleotide, right? Same thing with the protein. Do you have amino acid, amino acid, amino acid, nothing, 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 amino acid, amino acid, amino acid? No, we don't. Right? Why do we align? Is because in that quest, in that quest to uh, make sense out of this, for example, identify something similar, because you can, so that's, you say, I want to identify something similar, but it kind of looks similar, like this part A looks similar, part B looks similar, but there's a whole bunch of dissimilarity there. But I want to align the similar parts. So it's a mechanical operation that we perform. Then we put a bunch of gaps, put a bunch of dashes, just so that we can get all the parts that are aligned nicely. Nature doesn't align, right? Nature does not have nothing. Nature has a protein. We start moving things around and playing around with it in order for us to make some sort of a sense. And the sense is like, look for something with a different structure. Look for something, identify something unknown by the similarity of its sequence. Identify something that's unknown and just say, well, is this evolutionarily related? And I've already repeated this 2 or 3 times, but nature does not need to align. We have to align to make sense of how this is. Now, that's why that even there is no universal. Remember when we talked about that 40%, 60% from the olfactory receptors? I told you that there is no direct way for us to say that yes, 40% constitutes a family. It's an arbitrary idea. You can rationalize it, you can argue it, you can say, okay, this actually makes perfect sense, in kind of the afterthought, or you played around with it and said my sweet spot percentage is 40 for a family and 60 for a subfamily. But nature didn't say it has to be 40 for a family, right? So this is kind of that human intervention in order to make sense of everything. So, having said that, let's look at these alignment. That means, just like there is no, somebody could have easily, I don't know, the PI of that group could easily have said, let's make it 35 as a family and 75 as a subfamily. Okay, that means you would have had a larger number of families, because you're restricting that similarity to 35% as opposed to 40. But within the family, because you're restricting to 75%, you would have a fewer number of subfamilies, right? Or actually, you'd have a larger number of subfamilies because you were not clustering them together, because you're looking for such a big dissimilarity, or looking for such a big restriction on whether something should be similar enough to fall into a category of a subfamily. Okay. Now, similarly, when you create a gap, right? Remember, we said we need to create a gap. You have to ask yourself at a philosophical level, why do I need to create a gap? Am I just taking two things in two different parts that should never be aligned, but in order for my quest to align them, to make sense out of it, am I just throwing gaps all over the place? And that's one of the reasons why there is what you're looking at in front of you, and that is what is called a gap penalty. A gap penalty is not, is saying that you are now redefining what nature has already provided for you, which means that you have to be very careful in how you assign these gaps. And therefore they say it's like that art and that science, right? I can put a gap in here, but should I put a gap in here? Should I just put a gap to, right, and just to make sure that everything works in the way that I want it to be? But no, that's so therefore you have to have a gap penalty. Now, obviously nobody's going to agree on gap penalties, or somebody can come up with a gap penalty and somebody says, no, I have a more sophisticated way of putting a gap penalty in there. Okay, so there's some gap penalties here that you may or may not like to put, but again, so one is what's called the constant gap penalty. Okay, and even gap penalties, this idea of whether we should put gaps in there is an evolving concept. Somebody comes up with something a little more sophisticated, right? So for example, I'll give you an example. If let's say, for example, each of you gets a final grade, right? And I decide to curve it. I say, well, I have a certain curve, and I want to curve it between an A and a B minus, and well, I'm going to see how people cluster, and I don't want to fail anybody, so I'm just going to take the lowest. And then if I take the lowest as a B minus and the highest as an A, I'm going to have to distribute everybody else based on that foundational idea. Alternatively, somebody can come and say, let's do this differently. Let's not do that linear thing just by inspection, but let me give you a certain formula. Okay, and in that particular formula, that formula actually is ends up that mathematical formula ends up being the mathematical formula for an exponential curve. So what happens in that exponential curve is, if you say that the exponential curve is this way, remember it goes up or it goes down. You might say if somebody ends up on his or her raw scores on that part of the exponential, on that part of the curve, you can go straight down, or have, depending on the nature of the curve, have a very slight increase or a slight decrease, right? Again, this is an idea that basically will evolve, and then you take a look at it and say, well, that mathematical formula was not very good because too many people dropped down. And then you say, well, but the mathematical formula was created based on the people's scores. And then you say, no, let's keep it simple, let's just do it linear, or let's just do it by inspection. Okay, so a constant gap penalty is a gap penalty you have to apply. Okay, so for example, look at our sequences. You have got A, T, T, C, G, T, C, A. And I was like, I'm going to align that, and I'm going to align the T, and I'm going to align the C, A. And because I see these little points of alignment, you have to have a gap penalty, right? And so what is the score? The score means that you have a negative score for every time you put a gap. Okay, give yourself a negative score, and that is, there's one gap here, right? One gap here, so it's a -1, right? One gap here. Don't you don't worry about the number of gaps you have, as long as there's a gap. I'm going to give it a score of negative score of 1. So the alignment is AA, that's +1, TT, that's +1, and CA is +1 +1, that's +4. But I put two gaps in here. I don't worry about the length of the gap. That's what the constant gap penalty defines. So now my score, which was actually aligning 4 together, now my score has been reduced by 2. Okay, reduced by 2, and that is my penalty for putting gaps in there. Okay, now you take the same sequence and you say, now we have what is called a linear gap penalty. In a linear gap penalty, you say that the length, you didn't worry about the length, but here you worry about the length and say, you know what, I'm going to give a negative score every time you put every time you put a dash, every time you have a kind of a lack of a match. So you would say, okay, so 1, 2, 3, 4, that's 4. And then I have one, -1 here, and I've got two gaps here. So I've got -1, -2, -3. Actually, there's -3. There's three gaps here. So this answer should actually be 0. So I put a gap for every, right? And so that this is incorrect, but that's fine. It's just something that we have to remember. Now let's say somebody came up with something called as an affine or affine gap penalty. Okay, something again a lot more sophisticated. Now again, see, there is no there is no mandate from nature saying that this is what you have to do, right? You look at something and you say, well, let's be a little more sophisticated. Let's treat this as a science and an art, and say, I'm going to do this, 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 and this, and I'm not going to do anything else. Okay? So you have an affine gap penalty. Now, what is the affine gap penalty? The affine gap penalty says that if you if you start a gap, that's called a gap opening penalty. Okay, a gap opening penalty means you, like, beware. That's what it's trying to tell you. Be warned that if you even start a gap, the gap opening penalty should be 11. And here, when you say 11, and this is when I say, it means just for example, is beware when you open a gap, because you're going to incur your score is going to your alignment score is going to get reduced by -11. Okay, and then if for example you have that, okay, then you say, well, I need three gaps, so it's like, okay, then I'm going to give one. Again, all of this is arbitrary, right? And then L is the length of the gap. Okay, so, and then you could also say, well, okay, what am I going to do? But if I don't have gaps, then I'm going to have mismatches. So then you could say I'm going, this is actually negative. You could say I'm going to have a +2 for a match, +1 for a mismatch. Again, not a, so you could, so notice something, there's an arbitrariness and a randomness to how some of these things have arisen, okay? But that's still something a little more sophisticated, that you would have the gap opening penalty, which is -11, and then you would have, so that's the gap penalty, so then you would have your scores, and then you would subtract from there the gap penalty, and then you would get your final score, so your score would reduce. So it's like, again, now you begin to play this little dance where you say, I'm going to look at this and really decide whether that gap is worth putting in there, because the penalties are pretty severe. So if you take that idea, and let's look, for example, here's a reference where somebody gave a sample sequence, and you have a two protein sequences, right? So this is proline, arginine, uh, threonine. T is for threonine, E is for glutamic acid, I is for isoleucine, N is for asparagine, and S is for serine, right? And then here you have the same thing, PRT and EIN, and no S here. And here you have a tryptophan, a proline, and a serine. And the simplest thing would be, well, this is the match. But how do we come up with that score if there is an alignment? Okay. Now, in illustrating this problem, I'm going to jump a little ahead, but then we'll come back. Okay, I'm going to jump a little ahead, but then we're going to come back. So, but when we look at that, I'll explain what I mean, that part where I'm jumping ahead. But I think this is the right place to do this. So, typically based on the affine gap penalty, right? You say here is for this particular gap. Okay, for this particular gap, I have a gap opening penalty of -11. Then I have, and this is just the gap penalty, right? We'll worry about the matches and the mismatches later. And then, and again, every time the penalty is -1. So you have a gap extension penalty, and you're using this gap extension penalty because you've actually extended this gap by 3. So -1. And then you have the length of the gap, which is 1, 2, 3, -1, right? That's this formula. That's this formula. So here, you went for this particular thing, using this affine gap penalty, you have encountered a negative score of -13. Just a gap penalty, we'll do the rest of it later. Okay. What else? There is another gap here, right? Here, there is another gap here that you open just along this S. So that is -11. But now the gap extension penalty is one, right? Because it's 0 because you've not extended it. So then this second term is 0. So there the gap penalty is just -11, and the total gap penalty is -24, -13, -11. So that's -24. Okay. Now, though that's just a penalty, right? Obviously, you're looking for some positives here so you can actually get a score. Now, so we saw how we calculated that, and that's 24. Okay, fine, bit of animation there. And now, however, you actually found a gap, all the positives that came from it in terms of the matches is 32. Where did we get this 32? Why 32? Why indeed? Okay, so the things that, let's go back, the things that matched are PRT and EIN, right? And this is what is called the Blossom matrix. It's called the block substitution matrix. And we will see what the block substitution. So this is where you take families of proteins, right? Known families of proteins and specific blocks of those proteins, and see where there is, and so we know the protein, we know the function, we know the families, and then you look at where is one replaced, where is one substituted with the other, in what part of there. So the number of substitutions based on the position, you calculate what is called that is, so we have a sequence here, you calculate what is called the log odd ratio, and the log odd ratios are put here. Now, I'm throwing a lot of stuff at you, but don't worry, we will go back and look at each of these terms. Okay, so just for the time being, I'm just throwing in the terms, but we will explain this in greater detail. But I said this in order to illustrate the gap penalty and how the score is. So now you might say a proline and a proline aligned with a proline. So you can see here I'm tracking this Blossom matrix. A proline aligned with a proline gives it a score of 7. Okay, that's called a log odd ratio of 7. So that 7 is here, right, for this thing. Then an arginine, R is a short form for arginine, interacting with an arginine is 5. Okay, so you've got 7 plus 5 is 12. Okay, then a threonine interacting with a threonine is another 5. So that's 7 plus 5 is 17. And then we have no mismatches, right? We have taken all the matches. Either we have matches or we have gaps. Okay? So the next one is the glutamic acid. And the glutamic acid is a 5. Now you look at some of these are negative, some of 0, some are negative. That means the log odd ratio of like tryptophan being in the place of alanine is very small, almost negligible. That's why these numbers are small. But once we come to log odd ratios and block substitution, I'll explain this a little better. So this is 5. So that was 7 plus 5 plus 5 plus 5 is 22. Then you have isoleucine, I aligned with an I aligned with an I is 4. So that's 22 plus 4 is 26. And then you have an asparagine, N, aligned with an asparagine gives you 6. So 26 plus 6 is 32, and that's where you get the 32. Okay, that's the 32. So matches. Now, if you gave your mismatches and you didn't put as many gaps in there, your score would differ. But you understand the point I'm trying to make here is there is no preordained hard and fast rule as to what these are supposed to be. Okay, these are kind of artificial constructs based on, I wouldn't just say, somebody just came up and made up a number. No, that's not the point. They made up this number based on, by kind of inspection, by observation, by looking at the trends in terms of alignment, um, uh, why you should be very very aware that when you put a gap in there, what are the bad consequences of putting a gap where it doesn't belong. Can you live with a mismatch, but just to match everything I'm just going to put as many gaps as possible. That is kind of the philosophical foundation of why this is happening. So I've got the notification that the thing will end in 15 minutes, but let's do this. Let us keep going until we're kicked out, and then you know what to do. Sorry about that. Okay, so let's go back to the PowerPoint. And so now you know how you came up with this. It is pretty much done in its gory detail. And so basically the alignment score is 8. Other ideas, mathematical constructs, etc. I'm just going to give you two. There are other things, and this is what is called a distance. Now, a distance, we typically understand a distance to go from A to B, and it's 10 meters away or 450 miles away, whatever that distance. But the idea of a distance, and it comes from this mathematical idea to look at the distance between two sequences is you take a sequence, you take another sequence. How many steps would you need to undertake in order to, or how many things would you need to replace to make the second sequence look like the first sequence or vice versa is the distance, right? So again, if you've not heard of this, this is a completely novel idea for distance, right? So the minimum number of single edits is right? And so you have another example, the distance between two strings. And so for example, here are my two sequences, right? They're kind of the same, but there's some motion in them. And then there are two. One is called the Hamming distance and the other one is called the Levenshtein distance. Okay, so let's look at how the Hamming distance is. So the Hamming distance says line them one below the other and identify how many changes you would need. Okay, and so the Hamming distance doesn't do anything except says how many changes. So you see the ones that are the same, right? Here's the first difference, and here's another difference, here's another difference, here's another difference, here's, and then it goes down the hall, right? Down the line. And so you can see the asterisks show how many of these are really different. And so you would say the Hamming distance between sequence S1 and S2 is 12. Now let's see what does the Levenstein distance form methodology prescribes. So it says that it's okay to use gaps and then compare, because what happens is you saw that these sequences kind of look the same, but you know but but you know but one one mismatch here staggered all the other mismatches and gave the others mismatch. So what if you just kind of put a gap here and then and kind of reorganize it a little bit? So that's the Levenstein distance. And so with the Levenstein distance, let's rewrite it a little bit as added gaps. So now once you add gaps in very strategic positions, now you have a lot of matches, right? And so once you have the matches, now you basically in the gaps are also included as mismatches, and you see that you have changed it to to 4, and you have changed it to 4, and so now you would say, well, the distance is less. Now what does it mean? Goes back to the usual question, right? We have basically given this a mathematical score based on an artificial construct defined by Hammond, and then the score changes because Levenstein did something a little different. Okay. Now these don't come from biology; these come from mathematics. And again, if you basically just do a simple search across the Wikipedia for like distances of these kind, you'll find there's a few more. And again, it's like going from the constant gap penalty to a linear gap penalty to an affine gap penalty, which we then illustrated in greater detail. I mean, you take the sequence from the affine gap penalty and put it into the use a constant gap penalty, and you'll see what that score is. It's completely different, right? But again, aligning, because of the big thing of identifying structural similarities, functional similarities, evolutionary connections, it's actually very important. Okay, now what are the alignment, again, alignment types? And now we're slowly going towards kind of alignment as far as the biological constructs are concerned. So there's a local alignment. So let's say you have two big sequences. You focus on like certain regions are really well aligned, and you will focus on those, and works best when sequences are dissimilar. Global alignment is when the sequences are mostly similar. You can put a bunch of gaps and make sure that the alignment across the board is the best you can get, right? So when sequences are dissimilar, you don't have to worry about like aligning the entire thing. You just say, I'm just going to align the ones that are the same, and if the others are mismatches, they are mismatches. And then you have something called as a global alignment, and there's a publication for this, which actually combines global, which combines global and local alignment, right? So you focus on the local aspect, but overall you're still looking at the global alignment. Okay, now what are the different, now again, remember something, that when we study things in class, we are looking at very you know, kind of scaled down version. And I always work on the scaled down version and make it kind of mathematically, etc., a little more rigorous. And the reason I want to make it rigorous is because if you know the kind of mathematics, the kind of the hard grind that goes into working on scale down, which you could literally do by hand, at least you begin to get an appreciation for when you take two sequences, dump it into an NCBI resource and say, do the alignment for me, it does the exact same thing. Okay, except it's, it's computerized. Somebody's written a computer program to do it, so it's rapid, it's web based and things like that. But I want you to get you will never have to, some of the things that we're going to do in the next few slides, you're never ever in your entire life going to do that. But the reason why I want you to do that is to kind of gain a kind of a foundational understanding of what you need to do, why you need to do it, in order for you to appreciate what goes on when you use a resource to do it. Okay, so there are different alignment methods. One is many different ways in which this is done. It's called the dot matrix, which is a little simplistic, and you'll see why. And then you have something called as the dynamic programming, which is what's called the Needleman-Wunsch method, which is good for global alignment, and the Smith-Waterman, which is good for local alignments. And we'll see what those are. So let's focus on the dot matrix methodology, which is basically what they do in terms of pairwise comparison. So let's say you have two sequences, okay, and one of them is written like across, one of them is written like across, like on a row, and then the other sequence is written at the bottom. And then if the sequences are this, like the, if there is a similarity like A and A, then you put a star. And if A is not, there's A and the other one is T. Now there are no gaps or any of these things, you just write them as is, right? So let's look at two sequences, ATGCATT and ACTGATT. Now if you look at these sequences, you'll say, well, I could do this by hand, yes, but that's not the point. The point of this is to, because you're never going to take these two sequences and dump them into, a pairwise aligner, and certainly if you take my course that I teach in spring, you'll find that I actually have a you actually learn how to write programs to do these things. Okay. All right. So we've taken ATGCATT and wrote it in a row, and then I took the other one, ACTGATT, and wrote it as a column. And then what I do is I say, okay, A and A is the same, so I put a star here. And then this A and A is also the same, so wherever there is a match, I put a star. Okay? And then this gives me some sort of an idea as to how these comparisons occur. So what does that mean? Now, if the sequences were exactly identical, if they were exactly identical, then the diagonal, right, the diagonal would be fully populated. Okay, but obviously they're not. So you're going to see, yes, A here. There's a little box here which says that ATT and ATT are the same, right? ATT and ATT are the same. And then there are some other patterns here which, may or may not make any kind of sense to you. So we look at the exact same thing and it's been reproduced. But here you will see something in this ATGCATT. You'll see that here A and A match, T and T match, you'll see that there is something else going on here, right? That you'll see that yes, this there's not much in terms of similarity, but you see a certain pattern is that one sequence is pretty much very close, not exactly, but very close. Actually, it is the reverse of the other. See that? It's the reverse of the other. So what happens is in the real diagonal, you're not going to get anything, right? But the off diagonal, so you begin to get these little clues as to what some of these things mean, and these are the patterns that you get to see. Now let's look at this. Now in a real alignment, and this is a real sequence of the human zinc finger transcription factor. So basically you're taking that one and you're taking something else and you're aligning it. Now obviously this is a mess, right? So a dot matrix is very noisy, as you say, and there is no such thing as insertion. There's no such thing as gap. You just line them up, and you see the pattern you get. So you see certain patterns begin to, right? Now you see certain things like a lot of noise, right? A lot of noise doesn't make a lot of sense, feels completely useless. But in the overall scheme of things, in one, just as a from a take a step back and look at the view, you'll actually see certain patterns, right? For example, this box looks very similar. So there is a big, big, big similarity here, right? Backwards and forwards, and even some repeat regions. See, there are same repeat regions, and so that's a block, right? And then within the block, you're going to see some further blocks of even greater similarity. You'll see some other lines, very symmetrical lines here, like this symmetry with this, symmetry with this. You'll see little boxes here of similarity, little boxes here of similarity. So you're going to, you get kind of a sense of what's similar about this thing. But again, you're not going to get, because what might happen is if you actually did a pairwise alignment, you might just say, one gap, one if I put one gap, everything would stagger into place and this would be perfect, right? So it's kind of difficult, right? So you can see sequences, reverse, repeats, etc., which are part of this, right? And you can see like kind of but very broad from a, okay. I hope that was the last time for today. Okay. All right. So you can see there are some advantages. You get a bird's eye view. You get to see some patterns which show up. And again, I'll give you the next slide will show you a little better version of the pattern. So for example, if I have two sequences A B C D A B C D A B C D A B C D, so they're just these repeat. Well, hopefully that's the last time that'll happen. So again, you see the advantages and disadvantages of a dot matrix, right? Very noisy, don't make a lot of sense. And as I said, you could just say, well, if I really inspected this, I could just add one gap and everything that staggered would stagger and fall into place and give me a great similarity, right? So there are some disadvantages to this. And I'll show you but you'll see some patterns here, and this is just a made up sequence just to kind of to illustrate the point, right? So if I just made a made up sequence like ABCD, 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 and then the exact same identical sequence. Now when we line them up like this, you'll see that yes, I told you that the entire diagonal is going to be in place, right? Because it's the exact identical sequence. But you'll see that because you see it's a repeat region, right? You will see that all these other little pieces are also repeated, right? Right up to ABCD with ABCD. See that? And that tells you that there are some additional patterns here, right? Uh, it's not going to like you're not going to see anything on this side because it's not reversed, but again a certain pattern begins to arise. You can see here there is also something very interesting. So if I say A, B, C, D, E, blah, 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 you see that this entire diagonal term is correct. That is correct. But then you see that within a region, the EDCBA, so it's ABCDE and EDCBA, right? Now these are just made up sequences just for illustrative purposes, but you see that EDCBA is the reverse of ABCDE, and because it's reverse EDCBA, right, is here, you're actually going to get a little region. That means not all of it is reverse. If all of it was reverse, you would get this, and like the entire reverse would happen this way. It would happen this way, right here. But because only a region of it is reverse, you get it. So once again, if you look at enough of these, you begin to see certain patterns. But once again, we are not really doing anything so rigorous that we're going to say, well, we're going to really add gaps and we're going to see indels and things like that, and therefore giving us the best alignment possible. So this is it has its place, but obviously we couldn't use something like this because it would not work. All right, and this is the basically the last topic I'm going to discuss because we've already done a lot. So this lecture will go into two. This particular PowerPoint will go into two lectures. Okay, so what is dynamic programming? Now this will take at least 30 minutes of discussion. So dynamic programming is a method of solving a complex program. Again, Wikipedia. So the basic, simple, straightforward definition by breaking it down into a collection of simpler sub-problems. Okay, we'll see what that is. And in the case of alignment, we have what is called a recursive relationship, where recursive or recurring relationship. So it's like, well, I have a big matrix, a 10 seq by 10 matrix. So the question is, how do I complete that matrix? Okay, who's heard of a, let's say, did I use this? Who's ever heard of a Fibonacci sequence? Anybody heard of a Fibonacci sequence? Okay, thank you, Michaela. Anybody else has heard of a Fibonacci sequence? So a Fibonacci sequence, so let me go to our web page and see. So let's go to our definition of a Fibonacci sequence. That's too big. Oops, sorry. And you'll see why it is the sequence of nature. Okay, so let's say we start with the numbers 0 and 1. Okay, and that is actually enough to generate the rest of the Fibonacci sequence. Why? Because the third number, the subsequent number, is in a Fibonacci sequence, is the sum of the previous two numbers. Okay, is the sum of the previous. So 0 and 1 gives you 1, 1 and 1 gives you 2, 1 and 2 gives you 3, 2 and 3 gives you 5. So remember, when we go back here and we say that they're looking for a seed number, right? A seed number means that you have a starting point and the rest of the sequence or the rest of the matrix generates through a recursive method, because we have a certain formula that every sequence essentially does. And when I say sequence, I don't mean a biological sequence, a mathematical sequence, right? So for example, what is the mathematical, how do you write a formula for the mathematical sequence of every even number? Okay, so it's 2n, right? 2n, because it's 0, then 2 times 1 is 2, 4, 6, 8, those are even numbers, right? Alternatively, how about odd numbers? Odd numbers is really where it's 2n minus 1. So 2 times 0 minus 1 is minus 1. 2 times 1 minus 1 is 2 minus 1 is 1. So 2n minus 1. And if you're just adding 0, 1, 2, 3, 4, etc., you get all the odd numbers. Okay, so that is a particular formula. In this case, the formula is every number. Now, these are the seed numbers. You can't start from nothing. These are the seed numbers, and these are the seed numbers, and then every subsequent number is the sum of the previous two numbers. Now, there is a lot of value to it. Okay, so you can actually see that there is in nature, the spiral the spiral associated with a lot of living organisms like snails and things like that actually involves this radii, this radii of numbers, and it all arises from the Fibonacci sequence. Okay, so even here, for example, this spiral is found in nature. It's called the golden ratio and the Fibonacci sequence, and all of this. So it's not just adding the two, it's actually something a lot more valuable, that is, it's essentially foundational to nature, that this gentleman called Fibonacci actually figured out how that sequence is created. Again, this was just an aside to actually tell you how you begin with a seed in a Fibonacci sequence. The seed is 0 and 1, and then that's it. You can write it ad infinitum, ad nauseum, and it's never ends, right? So again, to have these recurring relationships, you start with a seed term and then you use the formulas associated with the recurring relationship to generate the rest of the matrix. In terms of alignment, etc., so then, and that is that methodology is basically going to come, is what is used. So identify scores for matching, mismatching, and gap penalties. So you say, okay, this is fine, but we still have to eventually, basically try to mimic what nature did. So you say, you assign a score. If there is a match, I'll give it a score of +1, +2, or whatever, arbitrary. If it's a mismatch, I give it a score of a smaller score. Obviously, a match score cannot be the same as a mismatch score. And if there's a gap penalty, we add a gap penalty too. Now, this is what the Needleman-Wunsch recursive relationship is. And now you might say, oh my God, this seems a little hard, but it does not have to be. Okay, so let's think about it this way. In a matrix, right? In a matrix, you have, let's say, let me show you. So in a matrix, for example, here you have, let's say, for example, let's look at this matrix. Okay, let me go here to study it out. I'm just making these numbers up because just to show you the point of what I'm trying to say. Oh my God, why is this so annoying? Okay, let's try something different. Okay, so here, for example, you will see that this is a 3 2 by 3 matrix. So the question is, as a convention goes, you ask yourself, okay, this one's nicer. This is what is called a 3 by 3 matrix. Okay, so 3 by 3 matrix means always look at rows. So 1, 2, 3 rows. Conventionally, rows are always written first, and then you have 1, 2, 3. So it's rows and columns. Okay, so this is row 1, column 1. So this is term 1 and 1, 1 comma 1. This is row 1, column 2, right? Row 1, column 2. So this is 2 comma 1, okay? 3 comma 1. Then this is row 1, row 2, column 1. This is 1 comma 2, etc., etc. And you can see that 1, 1, 1, 2, 2, 1, 2, 2, right? Where rows first and column is second, and that's how the numbers are given. So if you go back to our slide, you'll see that this is what it means. So T is the term in the matrix. So this is term ij. So this could be 1, 2, 2, 1, 2, 2. This is on the row 3 and column 4. Then this would be 3 and 4. So now you see what that ing. So it's not esoteric. It's not weird. It's not kind of all over the place. You have to understand this thing. Okay, so the recurring relationship is you start with a seed and then you use that particular formula, right? So let's say if this is, so then what you do is what is the term in the previous term? So let's say this is term 1, 1. I is 1, j is 1. And I want you to do this by hand, and there's a homework coming up, okay? So I, so then if I is 1 and j is 1, T, this is the term in the matrix. This is 1 minus 1 is 0. J minus 1 is, that means, remember, this is 1 and 1. So 1 minus, so this is the 0th term, the 0 column, 0 row, plus sigma. Now sigma basically just means that if it's a match, it's +1. If it's a mismatch, it's -1. If it's a gap, it means -1. Okay? And we'll see what that gap penalty, if it's a gap penalty, then if it's I minus 1, that means it's term 1 minus 1, so it's 0, and j is 1. So this is term 0 and 1. 0th row, now you might say, where did that come from? We'll see. 0th row and column 1 plus the gap penalty. And you say the gap penalty, I'm just going to keep it. We're not going to do affine gap penalty, any of that. I'm just going to -1. Again, all of these are arbitrary, right? And then so what happens is, because when you start with a seed like 0 0, then you have 1 1, and 1 1 comes from 0 and 1, right? And so therefore 2 and 2 will come from 1 and 1. So you see that every time you put a seed, that you're going to create numbers, the previous terms, and then those previous terms will contribute to the term, to the next term. And so all you have to do is start from a seed, and then you can generate everything else. And I'll show you step by step by step how that works. And again, when so for example, if you say I'm putting TGGT like in a dot matrix, you're going to put one above the other. TGGT, you'll say, well, T and A are a mismatch. Okay? Now, and therefore I'm going to give it a score of -1. Or if there's a gap involved, I might give it a score of -1. Now, what do I mean by max? So if you want to create term 1 and 1, I'm going to have to do this calculation, this calculation, this calculation, and then the one which is the highest, which gives me the highest number for these calculations, is the one I'm going to put in 1 and 1. Again, this might seem kind of hard right now, so I am going to do this step by step, and you'll see what I'm talking about. Okay? So that could be the map or the mismatch. So now let's look at a Needleman-Wunsch algorithm. So you see, this is not officially the matrix, so this is the 0th term, 0th row, right? 0th row, because it's not really counting, but the first column, second column. So remember what we said, TGGT and ATCG, okay? TGGT and ATCG. So this is a 0 0, this is 0 0, right? This is 0th row, right? The 0th row and the 0th column. So we have to put a seed here and then fill all these numbers up. So you'll see what I'm talking about. Okay, let me move me up here, because I have shown you the calculations, and the calculations will show you how it works there. And remember what else we have to do. We have to do every one of these calculations, and if you're told that this is a -1, add a -1 anyway, because that's going to be the gap penalty. Eventually that's going to show up when you do the calculation. Okay, again, seems kind of hard, but it won't have to be. Okay, so now again, row 0, column 1, row 0, column 2, row 0, column 3, similarly like that. Now, the way you do this, you say, I'm just going to put a seed value, and that seed value is going to be 0. Okay, this is 0. So this is the 0, T00, T00. Now, how do I complete this first term? Okay, so let's look at this calculation. So I want to put a term in 0, 1. So my I is 0, and my I is 0, and j is 1. Okay, so now remember our formula. Again, the recursive relationships are put here. So it's the max of 3 calculations. Okay, 3 calculations. So I look at max of, so remember, this is, the first case was I minus 1, and j, right? I minus 1, here was I minus 1, j minus 1. Okay, and you can kind of refer to it back and forth. And then you have the sigma. The sigma says whether 0 and 1. But 0 and 1, there is no match because it doesn't really exist, right? 0 and 1 would be here, so there's no match. So this term does not exist, so you can't use it. What about -1 and 1? Well, we only start from 0 0. We only start from 0 0, so that doesn't work. But T00, this is the third equation, the recursive relationship equation. So you have T01, right? 0 0. So here you have j, I, and j minus 1 will be 0 0. Now what is 0 0? We already created a seed. And we said, so this is 0 minus 1. And the gap, now remember, if you're confused here, look for those 3 recursive relationships. Okay, those recursive relationships are what you need, and the value here is -1. So now this does not exist, this does not exist, so we can't, because that 0, 1, there is no similarity or dissimilarity, it doesn't exist. Same thing here, the negative 1 and 0 does not exist. Negative 1 and 1 doesn't exist because it all starts from 0, 0, right? 0, 0, 0, 0 on this side. And this is the only one that exists, you're going to put a negative 1 here, right? Now let's look for the next term. The next term is term for row 0, column 2. Now in this case, you have, this is I is 0, j is 2, so I have I minus 1 is negative 1, but negative 1, the term does not exist anywhere here, right? So this term goes away. Same thing, negative 1 and 2, because this is negative 1 does not exist, but this is 0 and 2, so this is 0 term and second term, this is I minus 1 is 0, right? Because this is this is I, so I minus 1 is 0, and j minus 1 is 1, plus g, which is our gap penalty, which was negative 1, so that is negative 2. Similarly, let's look for 0, 3, 0, 4. Again, we have anything with a negative 1, because I minus 1 is negative 1 when it is 0. This is negative 3 and negative 4. Because, and again, now I calculated both together, so the other recursive relationship, because the negative 1, negative 2, and 1 do not exist, so that's how it is. Let's go to the next one. Again, this might seem a little hard, but follow it step by step and keep the recursive relationship. Now we're looking for the term 1 and 0. So if you look at 1 and 0, this is I and j. So the first term was I minus 1, j minus 1. So I minus 1 is 1 minus 1, 0. J minus 1 is 0 minus 1, negative 1. And negative 1 does not exist because it would be here. So it does not exist, right? And therefore we do not include it. And here you have I minus 1, j. So I minus 1 is 0. J is 0, plus g, which is a gap penalty, which is negative 1, and 2 minus 1. Anything where this is negative 1 does not exist, and therefore this is 1. Okay, similarly, we'll look at the exact same thing for this term here, and anything with a negative 1, again, look at the formula. It's I minus 1, j minus 1. This is ij. So you literally, it'd be nice if we just wrote it. I is equal to 2, j is equal to 0. So then this is negative 1, this does not exist. This exists, negative 2, this does not exist. And then similarly, these two are, and we'll do one more, and then you'll see how the others complete. So once you look at this, right, you do the same one. So now you have 1 and 1. Okay, 1 and 1, and then you have that, and you fill out all. So again, remember, we have formulas and we have a seed number, and starting from the seed number and knowing that some numbers, the t minus 1, the row 1 minus 1 does not exist, it does not um, exist, and therefore we don't include them, and we only include the ones that do exist, and you get these numbers. Now, okay, so now what? So again, if I went too fast on that, do it slowly, do it by hand, which is the homework anyway. And now what you have to do is you say, okay, now I created the matrix. Now how do I determine what's my gap and things like that, right? So what you do is then you start with the biggest number on the left hand. And you look, even if you, let's assume you did it by hand, right? You would say none of these really match other than the G, right? The G matches here. I could match the T here, which means I'm going to have to add a bunch of gaps. So what you do is, at the bottom right hand side, you look for the one which is the lowest number, the highest number, sorry. Remember, all these are negative numbers, so the highest number here is 0. And then from there, you take an arrow to the next highest number. Now see, if you look at the 0, negative 1, negative 3, negative 2, negative 1, 0. So you go to the next highest number. Now from 0, you go in this direction. You have a choice. You can go to negative 1, negative 1 or negative 2. Obviously, you're not going to go to the negative 2, you're going to go to the negative 1. Or now, you might say, but that how do I know for sure? You don't. And that is one of the cool things about, or the uncool things about this, is that there is an arbitrary aspect. Now, when you go to BLAST, when you go to NCBI, and you take it for granted, that program is actually making a determination like this. So I'm just going to choose it went up this way. So the question is simply this. Look, here is the 0, right? So it's ignoring this and putting a gap here, and see it. This is the highest number. Highest number means you're likely going to get a match. So this G and this G. Then what it's doing is, you could go here, which was another G and another G, right? But then you want to go either this side or this side. It decided to go here, which means you're matching that G with that C. That's what the program does. Then here it is easy. From here to here it's easy, because there's negative 1, and 0. You want to go to the highest number. And then at the end, again, this is also going to be a gap. So it can either go here or it can go here. And I just chose it here, but the program will eventually do it. So now what happens is, you got a G matching with a G. You got a G matching with a C, which is a mismatch. You got a T matching with a T, which is a high number. And then you've got an A, which has a gap in it. So you'll see that is the match you get. Okay, let's go back one. And you see the match here and the match here, the match here and the match. But when I say the match, even though there is a mismatch in your alignment, you're just going to put a G over a C in order to make sure that the T and the T and the G and the G match. And then you're going to have a gap for this T because there's no match. You're going to have a gap for this A because there's no match. And that's basically going to be your alignment. Okay? Seems a little weird, involves a lot of really hard work. Again, in between now, if you start your homework and you kind of get stuck on something, then come. But meanwhile, just try your best to do this. Now, if you want to find out more about the Needleman-Wunsch, there's like a 50 page slide share. Now, when you go to BLAST, depending upon the nature of your sequence, all of this stuff is programmed. But I gave you a small scaled version of it, so that when you go to the highest scale, you at least appreciate the programming that went on to actually make it happen. And here's a slide share. Now, the next one, and this is going to be your homework, and I don't have to give you the homework because I'm already sending you the slide, is that it's called the Smith-Waterman. And remember, we said that one was global and one was, that Needleman-Wunsch was local, Smith-Waterman was global. Oh, I'm sorry, this was local. So you see the recursive relationship, very similar, very similar, very similar. There's one difference, that the max is going to be 0. Okay, that means either 1, 2, 3, or 4, and you have to put in your matrix the one which is the highest. You're going to put into your matrix one which is the highest. Now, remember, in your Needleman-Wunsch algorithm, you had all negative numbers, right? Other than a few zeros, they were all going to be negative numbers. Here you're going to get every positive number, because your recurring number is going to be a 0, so your max. And remember, anytime there's a negative number in any of these, you're going to get a 0. Because remember, you're choosing the maximum out of these 4, and 0 is going to be the maximum. Okay, 0 is going to be the maximum. And therefore, you're going to get a lot of positive numbers. But once you do that, you're still going to do this stuff. Okay, you're still going to do this stuff. But it's slightly different because you're going to use a 0. So this will be homework for next week. I'm already giving you the answer. And while telling you there's an answer, I've actually made one mistake here that you're going to have to catch. You don't have to tell me there's a mistake. You just catch the mistake. So the answer is already available for you. Okay, the answer is already available for you, because I want to kind of boost this a little bit. And so again, this is the answer, but there's a mistake, and I'm going to take away some of this stuff in the but so the question is, if I'm giving you the answers, right? If I'm giving you the answer, the sequence is the same as Needleman-Wunsch, why am I making you do the homework? because I want you to see the population, and one of these is wrong. Okay, so just be aware that one of those I've made a mistake in one of these. I'm not saying that it would be disingenuous of me to say that I've deliberately made a mistake just to catch you. No, I actually made a mistake in the calculation, but I've let it remain in there. So if you, but I know what that mistake is. So when you find that mistake and you got to fix that mistake, the alignment is exactly the same because this is a small score, and -2 for a gap is what you might want to do. But again, the reason why you have zeros is because of your four recursive relationships, two of them are there is a zero, which means anytime you have something negative, you still have to get the highest of that lot, and zero is the highest. Okay, and so you've got the answer here, and then you get the rest of it is fine. But I want you to show me by hand the actual choice calculations that you've made. That's why I've given you the answer, not because my interest was in finding the answer, because you could do this by hand, right? You don't have to necessarily do it. But again, you have to understand that how does BLAST or a pairwise alignment or a multiple sequence alignment do this, and then eventually add gaps and things like that. Okay, I think this is important, and we can stop here. We are about 15 minutes short, but I think your brains are full right now. And let us again, I'm going to see if there is any questions in chat or anything else that you might have for me. Again, you want to make an appointment with Google Meets, etc. But again, don't just say I don't understand anything. Okay, I want you to try. You can also come by my office if you're interested in. If you get stuck, you don't have to. You don't have to show up, but if you get stuck on any of this, okay. So I'm going to stop here. I'll share the video with you. You know what the homework is. You just have to look at the slides, and I'm going to give this homework 20 points. Okay, so you have to give me something. Yes, Maylene.
So when we turn in this homework, is it okay if, or would you rather us do it on a piece of paper, or can we do it electronically, kind of like on an iPad, and then turn that into a PDF file
As
and turn
long
it
as
in?
it's legible, it doesn't matter if you slipped a handwritten thing under my door, or do it on a iPad like using a writing thing implement and then send me the PDF. As long as your calculations are legible, you can. Sometimes what people have also done is you write it on a piece of paper, take a photograph or a scan of it and send it to me. That's perfectly fine. But if you write illegibly, then that's going to be a problem, right? I mean, I want to be at least be able to read. If you
Right.
if you've lost the ability to write, like I have, because we are typing everything now, then that's an issue.
Okay, thank you, sir.
Yes.
And are you wanting the handwriting for both the one we did in class with the Needleman
No, no,
one,
just this one.
or just the Smith? Just
Just
the
the
Smith
Smith
one.
Waterman, yes.
Okay, thank you.
Any other questions? Okay, so this is a good place to stop, and then we'll finish the rest next time. And if we have a little bit time, we can actually start the next course. But again, typically the classical bioinformatics takes 2 lectures. Okay, very good. So thank you very much. Again, if somebody can send me a reminder by Thursday, and I'll try and get this, but I'll make the video for you and send it to you now. All right. Okay then. Take care.